Hello, budding biologists, and welcome back to your video notes. Today we are going to transition away from cells and we're going to begin our discussion on viruses. Let's make it make sense, shall we? All right, by the end of this video, you should be able to compare and contrast the structure of different viruses, compare these structures to cells, and describe the role of viruses in causing diseases such as HIV and the common flu. Let's begin by discussing common viruses. I encourage you to draw and label each of these common viruses as we go along, as well as write down the function of each structure we discuss. Our first virus is adenovirus. Adenovirus is important because it causes several common illnesses, such as respiratory infections, conjunctivitis, also known as pink eye, and gastrointestinal issues. It's significant due to its ability to affect both humans and animals. Please be familiar with the structure of adenovirus. This is the basic viral structure most people think of when they think of viruses. The first component you need to know is going to be the surface protein. Surface proteins go by several names, so if you hear the words antigen, glycoprotein, spikes, or surface protein, we're referring to the same structure. These external proteins are connected to the capsid, and they're important because they allow the virus to attach to the host cell in order to infect it. Similar to enzymes, viruses follow a lock and key model, and the surface protein is going to be the key in the situation, and then a receptor on the cell is the lock. Next is going to be the capsid. That's your next structure. And this is essentially a protective protein shell surrounding the genetic material. It plays a crucial role in safeguarding the virus's genetic instructions as it navigates the host environment. Which brings us to our third structure, and that is the virus's genetic material. Remember, our nucleic acids, such as DNA or RNA, are going to contain an organism's instructions for replication. It's going to be the same with viruses. The genetic material is vital in viral replication. All right, next we have HSV, herpes simplex virus. And HSV is important due to its prevalence, especially HSV1, which causes cold sores. It's estimated that 67% of the global population is infected with HSV1, many of whom do not realize they're infected. Regarding HSV, there are 4 structures you need to be familiar with. Similar to adenovirus, HSV has surface proteins. We're going to call them a glycoprotein for HSV, a capsid, and genetic material. For HSV, note that the genetic material is DNA, not RNA. HSV only has DNA. In addition to these components, HSV also has a viral envelope. This is a lipid bilayer surrounding the virus. It facilitates host cell entry and allows the virus to evade being detected by the immune system. All right, your third virus to be familiar with is the T4 bacteriophage. T4 bacteriophages infect bacteria. They play a significant role in bacterial infections and how we control those infections. An example of a type of bacteria commonly infected by phages is E. coli. And structurally, we're going to focus on the first 3 components: the surface protein, capsid, and genetic material. Note that for bacteriophages, we're now calling the surface protein spikes. Bacteriophages also have other structures such as a collar, sheath, base plate, etc., and these offer structural support for the bacteriophage. But regarding this virus, please focus on the capsid, genetic material, and spikes. Finally, we have HIV. Understanding HIV is crucial because it leads to acquired immunodeficiency syndrome, better known as AIDS. HIV consists of surface proteins again, here we're going to call it a glycoprotein, a capsid, and genetic material, just like adenovirus, but we have some important differences here. Firstly, the genetic material in HIV is only RNA. HIV does not have DNA, it always has RNA. Next, HIV has a viral envelope. This surrounds the virus and helps it enter host cells. Please note that the most important structure to understand regarding HIV is reverse transcriptase. Reverse transcriptase is an enzyme responsible for converting viral RNA into DNA through a process known as reverse transcription. Which brings us to our next concept, retroviruses. HIV is one example of a retrovirus. Retroviruses, like HIV, are unique because they can convert their RNA genome into DNA once they infect a host cell. This process is called reverse transcription, and essentially, when the retrovirus infects a host cell, it carries with it a single-stranded RNA genome. Our enzyme, reverse transcriptase, then binds to the viral RNA, and it starts the process of creating a complementary strand of DNA from the RNA template. The result is a double-stranded DNA molecule known as proviral DNA. That proviral DNA becomes integrated into the host cell's genome, essentially becoming a part of the host's genetic material. The reason this is so important is that this allows the viral genetic information to remain dormant for a long time within the host cell, escaping the host's immune system and any antiviral drugs it's introduced to. Over time, proviral DNA can become active, leading to the production of a new virus particle, or particles rather, and potentially causing disease progression like AIDS. This viral strategy showcases the universal nature of nucleic acids. So jot that down. Remember, it is a retrovirus. All right, let's talk more about those nucleic acids. Remember from our last unit, nucleic acids, our DNA and RNA, serve as the molecular basis of genetic information for all living organisms. This means that the sequences of nucleotide bases that make up DNA and RNA are used to store and transmit genetic information similarly, regardless of what organism they're from. When a virus infects a host cell, it introduces its own viral nucleic acid into the host's cellular environment. Once inside, the viral nucleic acid can become integrated with the host cell's own genetic material. This can be so seamless that it's challenging to distinguish between the viral and host cell nucleic acids, including viruses. DNA or RNA of all cells and viruses contain the same nucleotides, and that allows the viral nucleic acid to be used by the cell without detecting an immune response. All right, moving on to viral specificity. Viruses are highly specific in terms of the host species they can infect and the types of cells within that host species that they're able to target. This specificity is often likened to a lock and key mechanism. The lock and key model suggests that viruses, our key, have specific molecules on their surface that fit precisely into complementary molecules on the surface of host cells, the lock. This is going to be very similar to how enzymes worked back in Unit 2. It's important to note the difference between antigens and receptors. Essentially, the host cells have receptors that act as a lock, while viral glycoproteins act as the key, making them specific to one another. While viruses are highly specific, viruses, similar to living organisms, can undergo genetic mutations. These mutations occur as a result of random errors during the replication of the viral genetic material, and they can lead to changes in the viral genome. One significant consequence of viral mutations is the potential for changes in viral antigens. Remember, antigens trigger the immune response in the host, and when a virus mutates, it might alter the structure of its antigens, making it more challenging for the host's immune system to recognize and fight the virus. One example of how viral mutations can impact human health is swine flu, H1N1 influenza. This virus was initially associated with pigs, swine, but in 2009, mutations in the influenza virus led to changes in viral antigens. This resulted in a new strain of H1N1 influenza, which had a different set of antigens on its surface, and these antigens made it possible for the virus to infect humans, which caused a global pandemic. Speaking of pandemics, let's transition to cause of disease. All right, we're going to start with the word pathogen. The word pathogen literally means disease causing. So a pathogen is going to be any organism or agent that can cause illness. As kids, we often refer to pathogens as germs, but for this class, please use the term pathogen or infectious agent. Bacteria and viruses are both examples of pathogens, along with parasites and a few other organisms. Regarding viruses as pathogens, viruses cannot move by themselves. They're going to rely on their environment in order to move about. Some of the common methods of transmission include airborne transmission, which occurs when viruses are carried through the air in respiratory droplets produced by an infected person. These droplets can be inhaled by others, leading to infection such as the common cold or flu. Contaminated food and drink often happens when viruses from fecal matter contaminate food or surfaces that are later ingested. An example of that would be hepatitis A. That virus is commonly transmitted due to poor sanitation or hygiene. Some viruses rely on vectors or animals, such as mosquitoes or ticks, to transmit the virus from one host to another. The virus may replicate within the vector before being transmitted to the host. Examples of this would be Zika virus and West Nile virus, which are both transmitted to humans through mosquito bites. The mosquito serves as a vector of transmission. Viruses can also be present in various body fluids, such as blood, saliva, semen, or breast milk. Direct contact with these fluids can lead to transmission. An example of that would be HIV or HSV. In each of these methods, note viruses rely on external factors or carriers to move. Next, we're going to discuss options for treatment once a pathogen has led to disease or illness. While there are other treatments and medications, the primary means of targeting pathogens is use of antibiotics or use of vaccines. Starting with antibiotics. Antibiotics are medications used to treat bacterial infections. Anti means against, and bio, life. So they work by targeting and killing bacteria or inhibiting their growth. Antibiotics are designed to disrupt the formation of the cell wall, which protects the bacteria from their external environment. It's important to know that antibiotics do treat bacterial infections, but they do not and cannot work on viruses. The reason they don't work on viruses is due to the structural difference between bacteria and viruses. Viruses don't have a cell wall to target, and therefore antibiotics are not effective in treating them. Vaccines, on the other hand, are effective in preventing both bacterial and viral infections, assuming a vaccine is available for the particular pathogen causing the illness, though. Vaccines work by stimulating the immune response to recognize and fight off specific pathogens. Typically, vaccines contain weakened, killed, or fragments of the pathogen. When a person receives a vaccine, their immune system learns to recognize the pathogen's antigens. If the person is exposed to the actual pathogen later, their immune system can respond quickly to eliminate the threat before the individual becomes ill. It's important to note that vaccines cannot cause the illness, but can trigger an immune response. Vaccines can prevent either bacterial or viral infections, but antibiotics can treat bacterial infections. All right, to wrap things up, I want to ask you guys an important question. Based on what you've learned, are viruses living? All right, remember, in order to be considered living, something must have all 8 characteristics of life. So let's go through them. Looking at our first one, viruses do contain genetic material. They either have RNA or DNA. They also adapt, though mutations are random and not in response to their environments. And they sort of reproduce, but since they require a host cell in order to make copies of themselves, we're going to avoid using the word reproduce and use the word replicate instead. So based on these 3 characteristics, it's kind of foggy. So let's continue. Viruses lack organization. They're not made up of cells. They lack homeostasis. They don't maintain their internal environment. They lack metabolism by not requiring energy, and they do not grow in size. All right, so let's revisit our question. Are viruses living? Based on the fact that they do not have all 8 characteristics of life, the answer is no. They're not living. All right, viruses are not living. Write that in your notes, please. Okay, and I'm going to wrap it up here for today. We will continue exploring viruses in our next video over viral reproduction. But until then, please schedule time at tutoring if you're struggling with any of the information in this unit, whether it's cells or viruses. Otherwise, I'll see you guys in class.